Okay. Thank you very much for coming to our bioinformatics seminar today. Today we have Professor Tunca Doğan from Turkey in very different time zone. I really appreciate for his willingness to give a seminar here. I know Tunca for many years. He has been very active in bioinformatics. I met him several times in the conferences, and recently I see that he's publishing very good high profile papers, including I think the topic he's going to talk today. He's currently the professor in Hacettepe University, Turkey. And previously, he was a postdoc researcher at UniProt protein database team at EMBL EBI and also in Cambridge University. And he has a PhD in bioinformatics. And he has been working on really the central part of many important topics in bioinformatics. I'm really happy to see him here today. Okay, so then Professor Doğan, please.
Thank you very much. Hi everyone, I'm Tunca. It's wonderful to be among you today. First, my sincere thanks to organizers of this seminar series, especially to Professor Kihara for the kind invitation and nice words to introduce me and the opportunity to present our lab's work. I've had the chance to listen to some of the talks in this bioinformatics seminar series in the past, and I should say they were truly outstanding, both the research and the speakers. So I'm quite excited to be here today. Today, I'll talk about applications of generative AI in biology and medicine. I find the work done in this area lately highly interesting because generative AI in biomedicine is not actually a distant promise anymore, but tools many of researchers can develop and deploy even this year. In just last couple of years, we moved from can AI imagine a molecule or a protein to how do we validate, regulate, and productize what it designs. And that shift is changing discovery cycles and experimental design. So let's move on. Let me first introduce our lab. At the Hacettepe Biological Data Science Lab, which I lead, we develop and apply AI-driven methods to analyze biological and biomedical data with the goal of generating and testing hypotheses about life. And our work clusters into 4 tracks: molecular generative AI, computational drug discovery, systems biology, and biomolecular function prediction. In each of these projects, we design a new algorithm or method and released it openly, both the source code and a ready-to-use computational tool, so the research community can use it and extend their own work. And across these projects, we emphasize rigorous evaluation, interpretability, and reproducibility, so that the model output can translate into actionable insight for biomedicine and biotechnology. And today, I'm going to talk about 3 projects that we have conducted in our lab recently. Actually, 2 of them is still ongoing. One of them is finished. The first one is DrugGen, the second one is FlowProt, and the third one is Crossbar, under different topics. So let's start with the first one. So the title of this work is Target-Specific De Novo Design of Drug Candidate Molecules with Graph Generative Deep Adversarial Networks. And the system that we have developed as part of this project, we call DrugGen. And it has been a collaboration between 3 different universities in Turkey, which we led. And the project has been finished in the last 6 months, and we managed to publish it. So I'm just going to try to explain what have been done in this project. So let's start with the introductory part, the problem definition. Discovering novel drug candidate molecules is one of the most fundamental and critical steps in the drug development procedure. And with the advancement in technologies such as high-throughput screening technology, it is now possible to scan thousands of compounds simultaneously. Because what we try to do in the drug discovery, at the beginning of drug discovery, is given a target which is mostly a protein in the human body, finding a selective and efficient binder. In most cases, it's a small molecule, but nowadays we are trying to develop biotechnological drugs which are peptide-based, protein-based drugs. It's also possible. In this project, we focused on small molecule drugs. And then while we are trying to find the interactions between that selected target protein and the drug candidate molecule, the number of candidates is quite high. So it's not feasible to test them each and every combination of molecule and the selected drug experimentally. And due to these reasons, the cost of developing a single drug was estimated to be around 1.8 billion US dollars, and the process takes on average about 13 to 14 years. And physics-based or machine learning-based computational drug target interaction or affinity prediction helps reduce the amount of experiments by scanning the potential small molecule and target protein interaction on a large scale. However, they are restricted by the existing drug-like molecule records in chemistry databases, which is in the scale of hundreds of millions or billions in some cases. However, on the other hand, it is possible to design approximately 10 to the power 60 small molecules that differ from each other by at least one atom or bond, indicating the nearly limitless potential of the theoretical space of drug-like molecules. This is a huge number, but of course, none of these drug candidate molecules have ever been designed or synthesized yet. We only explored a tiny, tiny fraction of this space until today. However, in our lab, we are trying to develop different methods and models that can help, well, at least we hope that it can help the, increase the efficiency of this drug development and discovery process. For example, a few years ago, we have developed a model called Deep Screen, which predicts the interactions between drugs and targets. And today I'm going to focus on the drug design model called DrugGen. And another model that we have developed is language-based model called Cellformer, predicts the properties, drug-like properties of the molecules. So after the design process, this can be used for filtering. And also we had a few years ago developed another model called DrugApp, which is trying to predict the outcome of clinical trials, or we can say the approval of a certain drug candidate molecule against the selected indication. So let's go back to our project, the DrugGen project. Here, we are trying to solve the problem of drug discovery, finding an efficient and selectable drug candidate molecule using generative deep learning. These generative deep learning models have been developed to create new data points according to available probability distributions that we use as training data set. In other words, they select new examples from a high-dimensional distribution space that is only partially known. Generative adversarial networks, in short GANs, learn by pitting a generator module against a discriminator, as we see at the top of the slide. This is a framework that can synthesize realistic samples from noise. This idea underpins many modern generative systems, and it has been effectively used to create synthetic human face images, as we've seen here, that are called deepfakes in the media. And now it's possible to generate images, audio, and even realistic videos with more developed versions of these architectures. On the other hand, at the bottom, we have the transformer architecture built entirely on the attention mechanism, providing scalable sequence and graph-based representations, and have become the backbone of today's state-of-the-art foundation models like large language models. These models illustrate how pre-training over a large corpus of text plus fine-tuning on a specific task can transfer the performance, the accuracy to some specialized tasks. And the idea behind our work is to utilize these approaches and techniques to generate drug candidates, small molecules conditioned on target context, aiming for tractable and synthesizable compounds. And this fits a broader trend where deep generative models are reshaping the early stage drug discovery, because other research groups and the many different companies, especially startups, are following this path. And our National Science Funding Institute in Turkey, called TÜBİTAK, deemed this idea worthy and supported our project in 2021. In this project, as I mentioned, we proposed the system called DrugGen, and the system's first application as part of the project, the first use case, was the hepatocellular carcinoma disease, a lethal subtype of primary liver cancer. Here, a hyperactive signaling pathway in this disease is AKT-mTOR pathway. So modulating this mechanism is critical to develop new, probably personalized treatments. And our expert collaborators in this project has selected the ACT1 protein, which is a key enzyme in this pathway, and become the target in our use case application. Effectively inhibiting this protein might be the key to develop these new anti-cancer drugs. And in the final phase of the project, we released the developed system as a ready-to-use tool. Now let's go back to the project and try to understand over the workflow. We begin with the data processing part. We compile small molecule, target proteins, and bioactivity records and convert each sample into a graph-based representation. We encode atomic and bond properties as node and edge features and feed them to our model, which is shown in the second stage in the middle. This is representing the design and implementation of the generative AI model. In the DrugGen project, we combine a graph transformer with a generative adversarial network. And after training the model, we generate de novo candidates and examine their performance with competitive analysis. And finally, at the bottom, we present the downstream analysis and filtering steps. We early filter candidates using drug likeness filters like Lipinski and Weber rules to eliminate unsuitable compounds. We compare physicochemical properties and visualize the chemical space with dimensionality reduction to monitor the structural diversity. We perform molecular docking on the target protein and rank candidates using machine learning-based bioactivity predictions. For top candidates, we test binding mode and stability with molecular dynamic simulations, and we validate our computational results with in vitro experiments in the final step. We can think of this as a funnel starting from the large pool of candidates, the de novo molecule records proposed by the model, DrugGen model. We narrow down the set step by step to reach a target-specific, synthesizable, and biologically strong core list. Now let's walk through the DrugGen architecture. On the left, we have the generator module. It takes molecular graphs as input, nodes and edges with annotations, and encodes them with a graph transformer layer. Well, multiple layers, of course. And multi-head self-attention mixes the local chemistry with the global context. And at the end, a readout multi-layer perceptron proposes a new de novo molecule that should match the target profile. On the right-hand side, we have the discriminator. It has the same type graph transformer backbone, but is trained to score molecules as real or fake. The real example we use are the target-specific molecules curated from experimental bioactivity data. The discriminator's feedback pushes the generator toward chemically plausible target-aligned structures. And in short, we can say the generator creates candidate molecules and the discriminator critiques them, and they share the transformer learns the chemistry that links the graph structure to target activity. So we designed and implemented this model, and we conducted small tests, and we go back and according to results of these tests, we try to optimize the model. This went on like 1.5 years, and in the end, we came to a point where we were satisfied with the performance of the model. At this point, we compare the performance over different metrics against other models in the literature. And there are many metrics, of course, but two of the most important ones we can call are the validity and novelty. Validity is about the model producing molecules that can exist in our universe that is governed by physical rules. And novelty is about producing original new molecules that are different from our target data set. And we should score high in order to deem the model successful. But there is a trade-off between validity and novelty. If you want to increase the validity, then the novelty decreases and you start to produce the same molecules that already seen in the training data set. If you want to increase the novelty, then the validity decreases because the model starts to hallucinate too much and produce molecules that cannot exist in reality. So finding a good point in this trade-off was important for us, and we did our optimization to manage this. And after analyzing the model performance, when we reach a good performance, we ran a series of downstream analysis to pick the strongest candidates among generated molecules. First, we filtered the candidates using these Payne's, Lipinski, and Weber rules. So we retained compounds with drug-like properties and found that nearly 50% of the molecules passed these filters. And after that, we subjected the de novo molecules additional downstream computational analysis. In the first one, we visualized the chemical space using UMAP dimensional reduction projections using the max fingerprint of the new de novo molecules. And we also add this bit into this bit the real molecules, the real inhibitors of this protein, which are shown with the yellow color. And the drug-GAN generated de novo molecules are shown in blue color, which stay close to each other, clustered together in the two-dimensional space. And then we further analyze them and see that they are structurally similar to real inhibitors. They yet remain original. And in the second filtering step, we use molecular docking against a ACK1 protein. Several de novo molecules outperformed the co-crystal ligand, which is found in the protein data bank, with scores around -9 kilocalories per mole or better. The bar plots here show the comparison between mean docking scores across different methods, and drug-GAN showed strong performance there. And in parallel to this, we predicted the ACK1 bioactivity with a machine learning based method, our deep screen model in binary setting as active or inactive predictions. Deep screen is a drug target interaction prediction system we developed previously, and it uses the molecular images, pixel-based images as input and process them via convolutional neural networks to classify them as either active or inactive against the intended targets. And with this analysis, we found that 56% of the candidate molecules were predicted as active, supporting the model's effectiveness. And overall, we obtained a list of chemically reasonable, structurally diverse, and biologically promising candidates focused on the ACT1 protein. We then narrowed the pool via expert curation and selected 30 molecules. And molecular dynamic simulations showed that interactions in the adenine region of the protein and the overall protein conformation were preserved in exemplar compounds, indicating the physically consistent binding modes. And we performed experimental validation over 5 selected molecules. We selected 5 of them and synthesized them, chemically synthesized them. And in the first validation stage, we ran enzymatic assays and obtained the dosage values in terms of IC50 for molecule 1, we call it molecule 1, for less than 2 micromolars, which can be considered quite good. And for the second molecule that is shown here, less than 50 micromolar. And it is important to mention that these molecules are the raw molecules produced by our model. These are not optimized by medicinal chemists to increase the activity. So if we do that, then we can increase the activity by possibly by orders of magnitude. And in the second stage, we conducted cell-based assays over the hepatocellular carcinoma cell lines, determining the effective dosage over cancer cells this time. The table at the bottom shows the IC50 values for molecule 1 and 2 across multiple cancer cell lines. And the data support biological activity in the AKT1 context. And in summary, in silico prioritization, MD-based feasibility checks, and enzymatic plus cellular validation yielded stored candidates for the preclinical evaluation. And this is the end of the project. In summary, the study aims to automate the target-specific de novo drug candidate design, reducing the effort and cost in early stages of discovery. We developed a powerful model for handling the complex molecular data by combining graph transformer architecture with a GAN. And currently, in vitro validation on drug-resistant hepatocellular cancer cell line is still ongoing. And potential impact of the study can be summarized as follows. Together with the related work in the literature, this approach could enable the discovery of entirely new candidates compounds that are critical for treatment of many different diseases. And regarding the open science, the source code, the data set, and the trained models have all been made openly available in our GitHub repository, which the links are shown here. And we also built the drug-GAN web application, which lets you use the models without writing any code. With the interface shown above at the top, you can design molecules for the proteins included in this study and download these records in the SMILES format. By the way, in the later steps, we plan to add new proteins, the models for new proteins, and add this in our web app and GitHub repo as well. And lastly, our umbrella paper was published last month. So please see the article for further details if you are interested. And now I would like to thank my team members, drug-GAN project team members. As Hacettepe University Biological Data Science Lab, we handled the AI and bioinformatics analysis part, and the co-first authors of the article, Atabey, Elif, and Melih Gökay, I would like to thank them, and I think Atabey is among us here today. And also I would like to thank Ahmet Osman Hayriye and Dr. Ahmet Rifaioğlu for their work. And on the other hand, our collaborators in the context of pharmaceutical chemistry and cell molecular biology from Gazi University and Middle East Technical University, Professor Dr. Erdem Manoğlu and Abdurrahman Olgaç, Dr. Deniz Cansen Kahraman and Müge Öcal Demirtaş. And also, I would also like to thank our National Funding Institute, TÜBİTAK, for the funding. Now, I would like to move on to the second project. This is about ProtGen, which is an umbrella term we use for all our models developed, model development studies aimed at protein design this time, okay, using generative AI. This is a collaborative effort, again, between multiple universities, bringing together expertise in AI, bioinformatics, structural biology, and medicinal chemistry. This research is still ongoing, and the results haven't matured yet. So I ask you to bear with me in the upcoming slides as this work continues to evolve at the moment. So again, let's start with the introductory part. Proteins are the functional workhorses of biology, and their function is deeply connected with the 3D structure and their sequence. And designing new proteins opens doors to novel therapeutics, catalysts, diagnostics, and more. However, the sequence space is huge. Experimentally exploring this space is both slow and costly. That's why there is a need for data-driven computational models that can help us efficiently generate proteins that can be both stable and functional. And there have been hugely successful research in the near past and present time. A considerable portion of it comes from Nobel laureate Professor David Baker's research lab and related companies. For example, they developed RF diffusion. This is a diffusion-based protein generative model, and they tested it on the use cases of protein monomer design, protein binder design, enzyme active site scaffolding, and motif scaffolding. Later, subsequent versions of this model have also been developed and released. The diffusion models which have been used here in this study are a class of generative models that simulate the data generation process by transforming a simple starting distribution into a desired complex data distribution through a sequence of operations. And these diffusion models for proteins are trained to recover corrupted or noised protein structures and to generate new structures by reversing the corruption process through iterative denoising of initial random noise into realistic structures. In another study, Madani and others proposed ProtGen. This is a language model like the natural language models that we are using every day that can generate protein sequences with conditioning on generic protein superfamilies. The model was trained on nearly 300 million protein sequences from thousands of different families and is augmented with control text specifying protein properties. This model was a 1.2 billion parameter neural network that is based on the transformer architecture, which uses the self-attention mechanism. And in the later parts, of course, they developed and released the second and the third version of these models, which are larger and more successful. And as a use case, they generated artificial proteins fine-tuned to 5 distinct lysosome families. And the aim of our project is to develop a computational system that will perform the de novo design of new protein sequences and also backbones with specific functions using generative modeling and deep neural networks, and to design single domain level protein backbones and sequences, as I mentioned. And at later steps, the proposed method is being validated by analyzing de novo backbones or sequences with physics-based calculations in a use case study. And for this purpose, the precursor DNA methyltransferase, DNMT3 enzyme family is chosen as our target system. Why this system? Because DNA methyltransferase family of enzymes catalyze the transfer of a methyl group to DNA. DNA methylation serves as a wide variety of biological functions, and DNMT3 mediated DNA methylation plays a leading role in the execution of many critical processes, such as the regulation of transcription, embryonic development, genome stability, and more. That is why it is critically important protein family for biomedicine. And another reason we choose this family is that our collaborator group in this project, Karacalap from the IBG in Turkey, is an expert on this system, so it can be considered as a suitable protein for generative model validation. So the first model we developed as part of this project is called FlowProt. It uses the flow matching strategy for efficient protein backbone generation. And the logic is similar to diffusion models, actually. Diffusion models add noise and learn to revert it to generate the structure. They are easy to train, but their inference is expensive. Flow matching, on the other hand, learns the mapping from noise to data distribution directly. They focus on learning an invertible mapping between the data points and the latent space, directly modeling the flow of the data in a smooth and reversible manner. And this makes the training harder, but provides a much faster inference. And FlowProt uses the flow matching strategy for efficient protein backbone generation. So here we are trying to generate proteins in three-dimensional axis. And FlowProt outputs 3D backbone structures residue by residue. Unlike general purpose flow models or generative models, we steer the generation using a classifier to target specific structural units as protein domains. By the way, my PhD student Ali Baran Taşdemir is working on this project actively. So let's move on to the second part. In FlowProt, we use a frame-based representation, which is derived from a previous study called FrameDiff. Each residue's position is represented by its alpha carbon's coordinates in the Euclidean space, and the orientation is encoding using the Gram-Schmidt orthonormalization, which allow us to represent the local geometry. The model learns to predict the translations and rotations that will carry the residues on their final position. And we use a binary classifier trained to distinguish DNMT proteins from other protein families. It is inspired from the diffusion model's classifier guidance principle. During inference, we use the gradient of the classifier to score and adjust the generation steps, pushing output towards the DNMT-like structures. And now, information about how we evaluate our output. We apply a three-step process shown here. For each backbone that our model is generating, we create a sequence from these backbones using the protein NPNN model, which is becoming a standard in protein design, AI-based protein design. And then we predict the folded structure of these predicted sequences using the ESM fold model. And we compare the ESM fold predicted 3D structure with the original backbone that our model generated, FlowProt generated, using metrics such as the self-consistency based RMSD and self-consistency TM scores. We also measure the similarity to the human DNMT enzyme using models such as Progress and more. These are the tools that calculate the similarity between proteins by using learned latent representation of protein structures. Now let's take a short overview of our results, because this is an ongoing project. For each sequence length, which is selectable, we generated many samples. And by using the evaluation pipeline, we generated many different folds for each sample. And we represent average score for the top 5 samples per residue in the table, in this table, which I'm going to explain in more detail next. Our collaborators are testing the FlowProt's top results using both physics-based methods and also machine learning models, such as Chai-1 or AlphaFold models. Here we see the results about SAM binding. By the way, SAM is a small molecule acting as a methyl group donor in the methylation process. So its interaction with the protein is critical. So we tested it, and also we test the interaction with the DNA later. Here we use 2 new metrics to evaluate. PTM is the predicted TM score that show the confidence over the predicted structure, and also IPTM, the score interface predicted TM score. Okay, interface TM score show the confidence in the interaction with the SAM interaction region. Higher scores indicate better performance in terms of functionality of the enzyme, and our preliminary tests, one model reached the IPTM scores of 0.76, which can be considered successful, and top designs like this are to be analyzed further with molecular dynamics simulations. And we also analyzed the DNA binding capabilities of our samples in a preliminary manner, yet using again Chai-1 AlphaFold 3. we tested whether these samples can bind to the certain segments of the DNA, the target segments of the DNA. As an example, we see a backbone we call backbone 37 in the slide at the left-hand side, which can bind with DNA with an IPTM score of 0.72, and another one at the right-hand side, backbone 38, can bind DNA with a lower IPTM score. And these two samples can also bind SAM with the IPTM scores of 0.37 or 78 for the right-hand one. This means that backbone 37 on the left one can bind SAM with lower confidence but with higher confidence to DNA, and vice versa for the right-hand side. So these were our preliminary results. We are now at a later step where we sampled backbones with high scores in terms of both SAM binding and DNA binding. However, they were not ready to be presented yet, so I could not include them. So for now, we can conclude by saying that FlowProt is the first flow matching model guided by a classifier to generate domain-specific protein backbones. We are working at the domain level here. It's efficient, function-aware, and open to generalization. So far, we present preliminary analysis done by our expert collaborators, and we are trying to find strong candidates from a populated sample group. As the next step, we are planning to send these samples for the manual testing in the context of molecular dynamics for the DNA binding capability of the design. We are also preparing a public release of FlowProt with web tool and public weights release for other researchers to use. And the manuscript is in preparation at the moment as well. So now at the end of this project, I would like to thank my collaborators. This was a collaboration, has been a collaboration between 3 universities, which we led as the Hacettepe University Biological Data Science Lab. We also have the Dr. Ezgi Karaca and her team from İzmir Biomedicine and Genome Center, who is dealing with the DNMT-related physics-based analysis and structural analysis, and Dr. Abdurrahman Olaç from Gazi University Pharmaceuticals Chemistry Department, who is conducting the protein-ligand interaction experiments. And I would like to thank my students, Ali Baran, Seyit Semi, and other students as well for their valuable work. Now, let's move on to the final, the last project that I want to talk about today. That is Crossbar, a unified computational framework for heterogeneous biomedical data representation and large language model-driven exploration. And in this project, 4 of my students, 2 PhD students and 2 undergraduate students, actually, Bünyamin, Erva, Melih, and Mert, worked really hard and built the model and the tool that I'm going to present in the next few minutes. So let's try to understand the problem that we are trying to solve here. In biomedicine, questions we ask span many scales, from understanding how a single protein works to studying disease patterns across the entire body. But answering these questions in isolation doesn't reflect how biology really works, because everything is connected. Now, we technically do have the data to explore these complex biological questions, but it's scattered across, hundreds of different biological or biomedical databases. And realistically, it is not possible for a researcher to even know all these resources, let alone collect and integrate data from each of these resources manually. And that's why we need integrative databases to bring this information together in one coherent, accessible structure. And to represent this kind of interconnected biological knowledge, graphs are perfect solutions. They naturally represent complex systems by capturing not just the data points, but the relationship between them. Specifically, biomedical knowledge graphs let us model how genes, proteins, drugs, diseases, pathways are all connected, and this enables us to analyze the biology in a more integrated manner. To address this problem of fragmented biological data, we developed Crossbar version 1 5 years ago, actually. That was a system integrating large-scale data sets across domains like genes, proteins, pathways, diseases, and drugs. And users could explore these networks through an intuitive web interface. But the system was built on a relational database, not a graph database. So conducting knowledge graphs from queries required complex processing, which limited the scalability. Also, the data span was low compared to what we need today after 5 years. And to overcome the limitations of the previous version, we built Crossbar version 2, Let me give you a quick overview of the full workflow, and I will go each part one by one in detail in the following slides. So, first we start by collecting data from diverse biomedical sources, and then reusable adapter scripts automatically retrieve and pre-process each dataset for integration into our knowledge graph. This time we store everything directly in a Neo4j graph database, enabling more efficient querying and updates. On top of that, we developed Crossbar large language model, which lets users explore the graph by simply asking questions in natural language. Finally, we offer 3 different interfaces: Crossbar large language model for natural language search, a GraphQL API for programmatic access, and Neo4j browser for visual exploration. And now let's try to understand each one. We integrated 34 different trusted biological databases covering a broad range of domains, including organisms, genes, proteins, compounds, from databases such as DrugBank or Campbell, and functions, pathways from databases such as Gene Ontology, Reactome, KEGG, and diseases and phenotypes from databases such as HPO, Mondo, and even more. And to automate the data retrieval and integration, we employed Pipette and Biocipher frameworks, which was developed by our collaborator in Heidelberg University, Saez Rodriguez research group. And together, this setup supports a standardized knowledge graph construction and enable continuous updates to ensure that the system remains synchronized with at the latest releases of the source databases. In total, the graph has nearly 3 million nodes, 20 million edges, and since the system is designed in a modular and extendable way, we can continue to add new node and edge types over time. And all is stored in a Neo4j database. Each node and edge carries rich metadata, including references, cross-database links, and molecular properties. And lastly, we embedded this structure into a machine-readable vector using 10 different embedding models. These include protein language models such as ProtT5, ESM2, and chemical language models like Cellformer, and also others for pathway, domain, and side effects and embeddings. These embeddings are for enabling semantic search and downstream machine learning tasks. And to evaluate whether the relationships in our knowledge graph reflect real biological facts, we conducted multiple use case studies. I'm going to mention just a few of them. In this use case, our goal was to see if the relationship between proteins in our knowledge graph reflect their functional roles. In our KG, the proteins are connected to many other nodes, and we want to see whether this connection, these relationships makes biological sense or not. To test this, we generated embeddings for each protein node using random walks along the biological meaningful paths, and we used meta-path-to-vec algorithm to learn these embeddings, and then visualize the resulting embedding space in reduced dimensions, in 2 dimensions, using the UMAP algorithm. And each protein was labeled with one of the 10 functional categories, 5 enzyme classes as shown here, and 5 non-enzyme classes, and colored accordingly. What we found is that proteins from the same category, especially non-enzyme ones, tend to cluster together. So we have clusters of different non-enzyme families. And then, as you see here, this color coding is showing the clustering, suggesting their graph context indeed captures their functional similarities. And now that we have built this comprehensive knowledge graph, the next step was to make it easy to explore. Because interacting with knowledge graphs often require technical know-how, like understanding graph schemas or writing in specialized graph query languages, such as the Cipher language. To make this more accessible, we developed the Crossbar large language model, which, by the way, is available through this URL, and you can also scan the QR code to reach our system. Let's see how it works. You enter your question, okay, in natural language. The system uses a large language model to turn this into a Cipher query tailored to query our knowledge graph. And we support a variety of models such as the GPT models, Cloud, Gemini, Llama, DeepSeek, and Mistral. And you choose your preferred one and securely enter your API key to run the query. And these models are integrated via API providers like the most famous the ones like the OpenAI, Google, Anthropic, as you know. And the Cypher query is executed on the Neo4j database, and it returns the structured results, showing the relevant biological entities and how they are connected. And here we offer two types of search modes. In the direct search mode, as we see the video on the right-hand side, the graph is traversed to find the explicit relation between the biological entities. And this is ideal for questions with known links, like which drugs target which specific well-studied gene. Okay. Alternatively, semantic similarity search look for biologically similar entities using high-dimensional embeddings. Even if two nodes aren't directly linked in our knowledge graph, the system still can find meaningful similarities. This is especially helpful when working with novel or less studied entities, the relationship of which are unknown, where you want to explore new connections and generate your own hypothesis. So the structured data obtained from our database is then again given to a large language model, and it translated into a natural language answer. Here is an example that we see on the right-hand side. We ask a complex question: what drugs are used for obesity disorder, interact with serotonin, and also target proteins involved in ABC transporter pathway? Once we enter this in natural language, all of the steps I explained happen in the background, and the system returns both the query, cipher query and the information about the related entries in a human-readable manner. For this query, the system identified several drugs. The result is written at the bottom of the slide, which match all the conditions in the given question. Now let's look at how well Crossbar large language model performs on two key tasks. First, we tested how accurately it can generate cipher queries from plain language questions. We created a benchmark dataset of 50 natural language questions, such as find all drugs that decrease the expression of genes related to Duchenne-Becker muscular dystrophy. We ask Crossbar augmented large language models to translate them into correct cipher queries. And as you can see in this figure, open source models such as DeepSeek R1 and Llama 3.1 actually performed on par with commercial models like Cloud 3.5 and GPT-4 when backed by our Crossbar LLM system. So even if you don't have access to expensive APIs provided by these famous providers, you can still get strong results using open models with Crossbar LLM. And in the second test, we tested LLMs on a set of 100 true and false biomedical questions, such as the Agar gene is associated with acute monotonic leukemia, true or false. Here we compared how the model performed on their own, the large language model like GPT-4 performed on their own, versus when it is supported by Crossbar version 2 knowledge graph. And you can see the difference clearly in this chart, where the striped bars are representing the standalone large language models such as Cloud, DeepSeek, Gemini, GPT-4, and the Crossbar-backed versions, Crossbar LLM versions of them, shown in clear bars. When you compare them, you can see the difference. Standalone LLMs mostly stayed near the chance level, meaning they were basically guessing. But when augmented them with structured data from our KG, the performance increased dramatically, so which also means that hallucination amount is reduced significantly. And finally, we wanted to test whether our knowledge graph could be used effectively as input for deep learning-based prediction tasks, relationship prediction tasks. We trained a model that we called ProtHGT, which uses our knowledge graph to predict the unknown links between proteins and goal terms. This is basically protein function prediction. And the model is based on the heterogeneous graph transformer architecture shown here, which is designed to work with graphs that contain multiple types of nodes and edges. ProtHGT updates initial features of each node through multiple HGT layers and then updated representations and pass through a multi-layer perceptron to predict the association probability of each protein go-term pair. To evaluate the performance, we use the DeepHGT benchmark, which includes a time-based split so that the model is tested on proteins it hasn't seen during training. This is similar to the kappa experiment, kappa challenge. And as you can see in the performance bars, ProtHD colored in red actually consistently outperformed all the other models across three main gene ontology subcategories, which can be considered successful. Now we are at the end of this project, and the preprint is ready, and it will be available in one of the preprint servers in the upcoming weeks. And I would like to thank my students that work hard on this project again, who are Bünyamin, Erva, Mert, and Melih, and also our collaborators at Heidelberg University Saiz Rodriguez Group for their support. And while finishing, I would like to thank all of my students at Hacettepe University Biological Data Science Lab. We are a lab of nearly 25 people. And our partners in Middle East Technical University and collaborators in European Bioinformatics Institute, UniProt Database Group, and in Heidelberg University. And I also would like to thank you for your attention.
Okay, thank you very much for impressive list of methods. I knew I was expecting that you talk about drug gen, but I didn't know that you have so many state-of-the-art works. Everything is very impressive. So we have some time for questions. Any questions from the audience?
Dr. Tunca, this is Boran. I'm a assistant professor in statistics department, and very great talk, and I'm very inspired by your work. Thank you so much. I have a very naive question. I'm very interested in the second project, the prot gene. I think you mentioned you used the flow matching diffusion model rather than directly applying diffusion model to this task. Is there any intuition of using flow matching diffusion model, and how does it connect to the protein structure?
Yes, actually, that's a great question, and nice to meet you, Dr. Gao. Thanks you for your kind words and the excellent question. Actually, while we were choosing our architecture, deep learning architecture for the ProtGen project, we play around a bit with different architectures, and one of them was the language modeling, obviously because of their popularity, and the other one was diffusion based models. But when we try the diffusion based models at that time, not today but at that time, because the field is progressing quite quickly, but it was like 3 years ago when we experimented with already existing models like the RF diffusion. We found some problems with them while running their data in the context of training and sometimes in the context of inference. And then we start searching for alternative architectures, because diffusion models are great, and if there is a way to address their problems, we would be happy to use that. And then we found about the flow matching models, which have a more direct matching between the start point, the noising point, to the your target structure, the prediction. And then we start playing around with that, and we found that especially during inference, it is quite fast and accurate, but their training was more difficult. So that we experimented it a little bit, and when we try to train it with a very large corpus containing millions of different proteins, it was nearly impossible to train. If we had sources like thousands of GPUs, we would possibly manage to train, but we didn't have those huge sources. With our limited sources, it was impossible to train. So instead we try to focus on the specific family that we want to generate proteins for, the DNMT family, the enzyme family. And we take this family samples, the real proteins, of course, from the different databases, from PDB, from UniProt, and we collect them and came up with a data set of nearly like 50 to 55,000 proteins. And we run the training procedure with only this data set, and it was even better than our expectations. It manages to match to the distribution of the DNMT family, and it's not only mimicking the real DNMT proteins, but it is producing novel results. So when we checked the results, we found that our successful protein designs have the sequence similarity, protein amino acid sequence similarity of nearly 25 to 30%, sometimes 20% to the real DNMT proteins of human and other mammals. However, when we check the backbones are quite consistent. So it means that the model learned the probability distributional space of the DNMT family. However, the downside is you need to train the model from scratch for another protein family. So now we are trying to get rid of this necessity by coming up with a reduced data set that represents the protein functional space, and we want to pre-train the model using this reduced filtered data set, and then we want to fine-tune the model on the targeted protein family. So yeah, that's all I can say at this point. Thank you.
Oh, thank you so much. Really great. I think I learned a lot more from your explanation. And one follow-up question. Well, since you also mentioned that it could be like computationally very intensive to train such diffusion model, have you also considered like a score function-based diffusion model, which kind of like a balance between both?
Yes, it's the excellent question. Again, we haven't tried that actually. When we tried the diffusion models for the first time, it was like the 3 or 4 years ago. And then we were experimenting with the early models, and the RF diffusion was nearly first. The preprint of RF diffusion was there at the time, and nothing else in the context of protein design. And we go back and check the image synthesis models for diffusion-based models. And yeah, so afterwards we directly move on with the flow matching. So we didn't check.
Yeah, thank you so much.
Thank you very much for the question.
Okay, I found there were a couple of questions, 2 questions in the chat box. So let me start from the first one from Nirav. I know he posted it when you were talking about, I think, drug gen.
Mm-hmm. I can go back.
Okay, so all right, yeah. So his question is that, do you embed target protein information anywhere in the GAN architecture, the network architecture, or do you embed the molecules specific for the target, and by that you consider the protein target? Where the protein target information come into this network?
Okay, this is an excellent question. In the first version of the project, when we started the project, the aim was injecting protein structure information again in the graph format into this model, and then we were trying to use the transformer decoder model and then leverage the cross attention between the protein graph and the molecular graph. And we tried to navigate through the molecule space and direct it to the protein of interest with this architecture. That was our first design. And we tried to work this. We used just a single generative adversarial network. We built 2 generative adversarial networks that are connected to each other in a serial manner. We tried many different things, and we couldn't manage to work that actually, because generative adversarial networks are notoriously bad. They are unstable. Training, managing a training with these models are nearly impossible. They are really bad. And then the lead author actually of the study, Atabey, my PhD student, come up with the idea that getting rid of the protein structure from the model and only use the real molecules injected to the model, to the discriminator model, which are the known inhibitors of the protein. So there is an indirect injection of the protein-related information into the model, not a direct, but an indirect one. This time, we managed to stabilize the training and then build the model, run the model, optimize the model, and get successful results. So in the final version, protein features are not directly used inside.
Okay. Yeah. Thank you very much for explaining even the history of the development. So I go to the next question from Achit Gupta. So he's asking if we can use some confidential information. I think probably he's talking about crossbar, or can you, I have a specific confidential information in a company, can I input such information in the system?
In the system. Well, it should be answered, I guess, in the context of which project it is. Is it the DrugCam project? Because the DrugCam project, if it is about developing new drugs, designing new drugs, since we released the model, the checkpoints and the source code and everything, so a commercial researcher can take this and use their own data in their own local servers and then produce the result. This will be the same for the ProcCan project. It's not yet public, but it will be public. If the question is about the Crossbar project, we again release the data. It's possible to download data and inject their own data into the knowledge graph. It is possible. However, it's not possible to do it with our user interface in the Crossbar project. So we have a specific user interface, and the Crossbar LLM is answering the questions based on our data, our knowledge graph. So it cannot be included. Especially sensitive data cannot be included because we keep it our own server. So that would be another great way. We should work towards federated learning, federated databases in order to incorporate this maybe in the future sense. But for now, we are releasing everything so that a commercial researcher can get the data and do whatever they want.
Okay. All right. So then I see that question from Pranav. Pranav, I think can you speak up? He's a student in my lab, so I hope he can ask a question directly.
Yeah.
Okay.
So also thank you for your talk, Professor. Really insightful. Lots of amazing tools from your lab. But regarding to in terms of Crossbar, I was curious about the knowledge graph construction. So in all of these databases like UniProt and etc., we also have like rich text information. So I was wondering if some nodes and relationships are extracted from the text information itself.
Yes, it's a great question as well. At the moment, we are not getting the free text information directly from these sources, but we get that information in a different manner because in the sources like UniProt, these are also added as metadata in a structured manner so that these UniProt flat files have many different fields, and on those fields you see a cross-reference, an ID pointing to another database, or for functional terms gene ontology annotation in the context of geo term IDs, things like that. We take them, all of them, and then put it inside node features. So this metadata is included as node feature in our knowledge graph, and it is queryable. So it is possible to ask a question like, I have a Go term that I'm interested in. This is the Go term, blah, blah, blah. Now find me the proteins that has this Go term, and also bring me the proteins that are interacting with the proteins that has this Go term, things like that. So this metadata is also part of the system, but not the free text information yet. But we are going towards this direction for probably in the third version.
Okay, yeah, thank you very much. Any other questions? All right. If not, since it's time, let's thank to the speaker again. Thank you very much for a really excellent presentation.
Thank you very much, Professor Chiara, for inviting me again. It was a pleasure to be here among you, present our work. Thanks.
Yeah, thank you very much.